Hi, we've talked about transcription, that is the writing of the DNA code to an RNA copy. Now we need to get down to business and make a protein. We call this process translation. If transcription is copying the base sequence from DNA to RNA, it's at least in the same language, the language of bases. Now we need to convert to an entirely new language. We need to make those base sequences into amino acid sequences. That's why we call this process translation. You already know the RNA code is written in words 3 bases long, called codons, and that these words stand for amino acids. There are also words that stand for start and stop. The RNA goes out into the cytoplasm and binds to the ribosome. Now we need to introduce a new character to our drama, tRNA, or transfer RNA. Many of you are already aware that there's more than one kind of RNA. There's the RNA that makes up part of the ribosome. There's the mRNA, or messenger RNA, that is the RNA copy of the DNA. And there's transfer RNA. tRNA transfers the amino acids to the protein we're building. But another way to remember what it does is to think of each of these tiny molecules as a specialized little truck that can only carry one kind of amino acid. Each tRNA has an anticodon. It's a strip of 3 bases that can bind to a codon on the messenger RNA. Once the messenger RNA that we made back in the nucleus is bound up to the ribosome, the transfer RNA's complementary anticodon can bind up to the messenger RNA's codon. On the other end of the transfer RNA is an amino acid, the only kind of amino acid that particular transfer RNA can carry. Once it's bound to the messenger RNA, it can release its amino acid, and then the ribosome binds to the next codon in the messenger RNA sequence. As the ribosome moves along the messenger RNA, the transfer RNAs bring in the appropriate amino acid to each codon and release them into the chain of amino acids that we're building. New proteins are complicated. They aren't just chains of amino acids. You already know that the shape of a protein is super important to its function. Our string of amino acids is unlikely to be the appropriate shape right out of the ribosome. But that's all right, because it will become the proper shape shortly. The first thing that happens is that the string of amino acids spontaneously arrange themselves. It can do this because some amino acids like to be in water and some amino acids don't like to be in water, and some amino acids are attracted to each other and some amino acids are repelled from each other. And because of all of these chemical reactions, the string of amino acids will spontaneously take shape. And then afterward, enzymes will come along and tidy things up and adjust bonds and link different units together. Eventually, after the string of amino acids has spontaneously shaped itself and the enzymes have come along and cleaned things up and attached subunits together, you have the perfect protein. And it can go out and do its job and be a hemoglobin or a muscle protein or whatever it is the cell needs. So here are some things to take away from this. DNA is copied as RNA in a process we call transcription. Number 2, RNA is converted into protein in a process we call translation. The ribosomes bind to the messenger RNA, and transfer RNA brings the appropriate amino acids into the ribosome to make the protein in the right sequence. And finally, proteins form their final shapes with the help of chemical interactions and other enzymes. Stay awesome. I'll see you next time. Young lady, are you finished?